Today we are having a little bit of a session on dominance, codominance, and sex-linked characteristics. I hear that these three and the interaction between them and what it all means is causing a bit of confusion. So let's make sure we're completely crystal clear. So dominance, codominance, and sex-linked characteristics. So when we talk about them, and we talk about genetics, and we talk about heterozygous and different versions of alleles, this is what we're sort of background to this. When we come to dominance, it's our sort of basic starting point. It's that straightforward of one allele expressed. It's our capital letter and our lowercase letter, and the capital one is the one that does the phenotype. Okay, that is straightforward dominance. When it comes to codominance, things get a little bit more interesting. In codominance, we actually have more than one allele expressed. So we can say here that both alleles are expressed. We are going to work through examples of all of these so you see them in a bit more detail. Okay, so both alleles will be expressed. So if we have a gene for coat color that is white and a gene for coat color that is red, we would have both red and white hairs through the coat. This is what we're going to look at in terms of cattle color. Okay, something to bear in mind here is that there's another kind of dominance in here called incomplete dominance. And that is when our two alleles are combined into one trait. But that's not part of the IG syllabus. So it can be good to be aware of it. But we often find that we just work with codominance within the past paper questions. And sometimes it might be a little bit dodgy because you might think actually what they're talking about here is one combined trait rather than two traits being expressed. But we're pulling it all back into sometimes dodgy examples of codominance. So dominance is when one allele is expressed. Codominance, more than one allele is expressed. Sex-linked chromosomes, sex-linked characteristics. I mean, obviously, chromosomes are sex-linked because we have X and Y chromosomes. Sex-linked characteristics, they have a different thing going on here. Because remember we said that the dominant allele is the one that's expressed. But when it comes to a sex-linked characteristic, you can have expression of that recessive allele because we have a difference between our X chromosome and our Y chromosome. So here we can have expression of the recessive allele without being homozygous recessive. Okay, so you can be heterozygous and have expression of the recessive. So what we're going to do now is we're going to actually focus in on each one of these and have a look at examples, and so that we can actually see them in action, and hopefully that will help. I'm going to start off with dominance. So setting the stage for that basic how things work. Sort of what is a standard. So also remember, we're saying here that there's two alleles that control one major characteristic. And life is far more complicated. We're simplifying a lot of systems to make this work. But it gives us a good understanding of what's going on and how genetics leads to an expression of a phenotype. Okay, so let us talk about peas and pea plants. Okay, when it comes to peas, we can have either yellow seeds or green seeds. So we can have, oh, I don't think that's not nearly, okay, not nearly dark enough to be seen. So we shall use ugly green for yellow seeds, and we can have green seeds. Now, if we put some genetics on this, we would have a capital Y for yellow and a little y for green. So that tells us straight away that this allele for yellow seeds is our dominant allele. And this allele for green seeds is our recessive allele. So what are the different genotypes that we could have? Okay, for pea plants, we could have 2 capital Ys. We could have a capital Y with a little y, and we could have 2 little y's. The thing to remember is that we're always going to have these in pairs, because remember, you get one from dad, one from mum, and that makes your pair of homologous chromosomes. Pair of homologous chromosomes means that you have one allele from dad, one allele from mum, and then they work together to create that trait. So we have yellow seeds alleles on both chromosomes, so we're going to get yellow seeds. And this is what we're going to call homozygous dominant. We have 2 capital letters. This one here is more interesting. See, we've got one of each. So mum gave us a yellow seed allele and dad gave us a green seed allele. But the thing is that we've said that yellow seeds are dominant. So as soon as we have that capital Y, what we look like is yellow seeds. And this is a heterozygous situation, but with straightforward dominance. So it just genotype, heterozygous, phenotype, yellow seeds. It's only when we get this situation, 2 recessives, that we can actually see green seeds. So this is our homozygous, both the same, recessive. Now we're going to see that trait. Okay, so if we then put this together and we have a look maybe at a Punnett square, because that's a great way to see. Let's start off with, actually we're only going to do one of these. Okay, so there's our Punnett square, and we're going to work with that fun combination where both parents are heterozygous. So both of the parents look like they have yellow seeds. But what happens when we cross them? Then we have 2 capital Ys. Here we would have a capital Y and a little y. I don't know how they turned into G's. We'll fix it now. And here we have a capital Y coming down from the top. So let's just turn those back into little y's. So they would come down here, this little y comes across here, and this one is 2 little y's. Okay, so now, when we look at our genotype ratio of our offspring, we can see that we have 1 homozygous dominant to 2 heterozygous to 1 homozygous recessive. Okay. If we look at the phenotype ratio, you see that we have 3 yellows to 1 green. And what you're going to see is, as we do each of these different situations of dominance, is that these genotype and phenotype ratios are actually going to change. So this is our standard situation when we have our capital letter, our lowercase letter, straightforward, dominant and recessive. Now we're going to move on to looking at codominance. What does that mean and how do we actually look at it? So codominance, we said, is that when we are expressing both alleles. So we no longer have the dominant which is expressed if it's there. We have 2 alleles which have equal dominance. So let's go back and talk about those roan cattle again. We're going to be interesting a little bit here as well. So when it comes to this special cattle, we can have red hairs and we can have white hairs. Goodness gracious, how are we going to do white? We'll do it in grey. Okay, but we have a different way of writing it already from the beginning when we're dealing with codominance. For codominance, we would call it with a C. For color, why not? Then we would put a little R or a little W. Now you see, both our R and our R and our W are capital letters. And remember, we use capital letters for dominant. So here we have two dominant capital letters. They are codominant. So what's going to happen is, if they are both there, they are both expressed. So if we've got three different options again. Okay, we're dealing with two alleles. So let's get our C's in there. Three options for color. If we have R, what are we going to have? We're going to have a red cow. Okay, and he's homozygous. If we have WW, we're going to have a white colored coat. Also, homozygous. See, we're not saying homozygous dominant or homozygous recessive here because there is no dominant and recessive. They're both dominant. They are both expressed. And that's where it gets fun. If we have one R and one W, so now we have the heterozygous, we actually get something different. We get red and white. So you'd have one red hair, one white hair, one red hair, one white hair. And when you mix that all together, this is what gives you a roan colored coat. So this now is our heterozygous. So neither the red nor the white is dominant. They are both expressed. So it can happen in carnations as well, where you have red carnations and white carnations. Now you get pink carnations. So that is when we're varying a little bit too far in the direction of incomplete dominance, because there the red and the white are mixing. It's not red and white spots making pink. Okay, so codominance is red and white hairs. But they often use that flower example for codominance in your exam papers. So just keep an eye out for it. Remember, if you're expressing both, then you're doing codominance. Okay, we don't have a recessive in this situation. Let's look at what happens if we do a cross with two roan cattle. For the fun of it, because that's our two heterozygotes, like we looked at last time. So we'd have two C's in each of these. Then we'll go back and fill in the other ones in color. So, two heterozygotes. So here we would have the R from the top, the R from the top, the R from the side, the R from the side. Okay, now the white allele. Then we bring it along the bottom and down from the top. And now you see we've got something a little bit different to last time. So if we look at our genotype ratio, we actually have the same. We have one homozygous to two heterozygous to one homozygous. But when we did the plain dominant recessive, we had a phenotype ratio of 3:1. But here, because we have codominance, that heterozygote actually has a different phenotype. So we have one red to two pink cows to one white cow. So our codominance really affects the phenotype. So we're looking now at describing our alleles with color, but two versions, red or white, rather than that dominant recessive capital lowercase letter that we used. Okay, then we have our different combinations of homozygous and heterozygous, and it affects our genotype and phenotype ratio because we are expressing everything. The other big example of codominance is your blood types. So let us talk a little bit about them because they are slightly different again. Remember we said it's a very complicated story, genetics, and we're simplifying it to get a grip on it, to get to know it a bit so that we can then take it further if we decide to take biology further. You can't learn everything all at IG. So when it comes to blood types, we have another similar story where we have codominance going on. So we have IA, IB, and IO. And you see, that's the sort of indication. We don't have our genetics of big A, little a, big B, little b, big O, little o. Okay? It is, we have an I with an A, an I with a B, an I with an O, and they are lots of capital letters going on. So we're still talking codominance here. Okay, when it comes here, we have slightly more interesting than our cattle because these guys are codominant. And poor little IO is recessive. So now we've got even more different options than we're considering. So let us think about the possible genotypes and phenotypes that we could get out of all of these. And let's get them in different colors, actually. So it's a little easier to see. Okay, so A shall be purple, B shall be pink, and O shall be blue to make him really obvious that he's different because he's recessive. So our genotype, let's think about two different options here. We could have A, O. Okay, next two options. We could have B, we could have B, O. We could have two more options. We could have A, B. We could have O, O. So now you've got to go through and you've got to think, okay, for each of these genotypes, what would my blood type be? So A and A. So we are blood type A. A and O. Now remember, the O in this case was special because he's recessive. So this one would again be blood type A. The next one, B and B, homozygous, straightforward. Blood type B. Next one, heterozygous, B, O. O is recessive. So we have again, blood type B. A, B. Those are both codominant, which means they're both going to be expressed. So here we would actually have blood type A, B. And our last one, homozygous OO, so homozygous recessive, we would have blood type O. So we've got a lot more variation going on here. So we're going to have to pick some of our ones to do a cross with. Okay, so let us pick some heterozygotes again. Maybe we shall go with an A heterozygote and a B heterozygote. So in that case, we would have to have an A, O, heterozygote, but blood type A. And then we can have a B, O, because now we've got a heterozygote, but blood type B. So what would be the fun and games if these two had children? Well, all of them would have 2 alleles. And then we can fill in. This one would have an A, and this one would have an A. This one would have a B, and this one would have a B. This one would have an O. This one would have an O. And then coming across, we'd have an O and an O. And look at that. We've again got pattern. Okay, so genotype, we've got 1 heterozygote codominant. We've got 1 heterozygote B, 1 heterozygote A, and 1 homozygous recessive. So again, a different pattern. Phenotype. We have AB codominant. We have B dominant. We have A dominant. We have O. So in this case, our genotype and our phenotype ratios are really boring because each thing gives us something specific that's different. So remember, when it comes to codominant, we can have expression of more than 1 allele. In this case, we have an interesting situation because blood types, we have 2 that are codominant and then the ones that are recessive. Okay, but it works the same for that A and B. When you come across them, take codominant. When they mix with the recessive, then they act just like a dominant allele. And when our 2 recessives sit there together, then we have that expression of that recessive trait. Where it then gets slightly different, okay, we said that we're going to be talking about expression of that recessive trait, even when it's not homozygous recessive, and that's in our sex-linked characteristic. So let's have a look at them. So sex-linked characteristics. Here we have to look first at the structure of our chromosomes. Remember that you're going to have 2 chromosomes, okay? 1 from mom and 1 from dad. But these are homologous chromosomes. Okay, you have 2 alleles for everything. So if there is an allele here for something, you have an allele for the same trait on the other one. And that continues all the way down. If there's an allele here, there's an allele here for the same trait. And even down in this little piece at the bottom, we would have same traits on both chromosomes. The problem comes when we look at our sex chromosome, because it works. I mean, this is beautiful. X and X, they're the same. We'd have homologous chromosomes. The problem comes when we get to an X chromosome and a Y chromosome, because a Y chromosome is shorter than an X chromosome. So now you have this whole region here of the X chromosome where there is no homologous piece. So that means that whatever allele that is on your X chromosome in that area, it's going to be expressed. Doesn't matter if it's dominant, doesn't matter if it's recessive. There is nothing to counteract it. There's nothing to balance it. There's nothing to fight against it. It's going to be expressed. Okay, so in this region, your alleles are expressed even if recessive. And that then links on to things that we talk about. So things like colorblindness is sex-linked because the gene for colorblindness sits at the top of this X chromosome. Hemophilia is another one that sits at the top of this chromosome. So we can say that hemophilia is an X-linked disease. So we would have another way of writing it. Because here, it's all about that X chromosome. So that does have capital H for normal, So normal clotting factors in your blood, and a little h for hemophilia. So now we have a blood clotting problem. Now we've said that that is X-linked. So on the Y, there is nothing to write. So when it comes to understanding how this would look in terms of different genotypes, we have various different options. We can have X, 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 3 different options for the women, but we only have 2 different options for male. So we could have a normal female. So she is homozygous normal. We could have H, little h. So here we are heterozygous. And the thing is that this woman is not going to have hemophilia because she's got that normal allele. So we have a normal dominant recessive relationship going on here. Capital letter, lowercase letter. So here we would call her actually a carrier female because she's carrying this allele even though she's not expressing it. Okay, then if we had little h, now we've got we've got homozygous recessive. So here we would have a hemophiliac female. When it comes to the XY combinations, nothing again on the Y because there's no region for it. So we could have X with a capital H being a normal male, or X with a little h being a hemophiliac male. So there's nothing to sort of counteract that. We can't have the heterozygous if we have a male because the Y chromosome doesn't carry this gene. So if we then take this and do our Punnett square, so we have a male, we have a female. Then we have, let's make it a carrier female with a normal male. So as far as they know, there's no hemophilia. Okay, then they have babies. Okay, 50% boys, 50% girls. But here's where it comes in different. These two get the normal gene from the mother. These two get the hemophilia gene from the mother. The 2 daughters get their father's normal genes, whereas the 2 sons just get his Ys. And now we have that thing. So XH, XH, all capitals. Here we have a female who is normal and she's not going to pass on hemophilia. Here we have a normal boy. He's got his normal clotting factors and his Y. Here we have what we call another carrier female. She's not going to have hemophilia, but she's carrying the gene. And this son here is a hemophiliac son. So here we've gone from that original capital and a lowercase, and that's our 2 plain simple dominant and recessive. Then we looked at codominant, where we used 2 capital letters, so we used like a C for color, and then red and white, both in capitals because they were codominant. When it comes to sex-linked characteristics, we're using our X and Y because we're talking about our X and Y chromosome, but then we're adding a little detail onto the X only because only the X has that region of expression for that particular trait.